Hello students, welcome to EPG Pathshala. I am Dr. Gunjan from the School of Biotechnology, Gautam Buddha University. Currently I am working as a assistant professor and head of the department in the School of Biotechnology, and my area of specialization is plant stress physiology and plant biotechnology. Today we are going to discuss about a module, Introduction to Agrobacterium tumefaciens and Rhizobium rhizogenes, under the paper Plant Biotechnology and Crop Improvement. The learning objective of this module are Introduction to Agrobacterium tumefaciens and Agrobacterium rhizogenes, which are now called as Rhizobium rhizogene. Then we will go to the characteristics feature, in what way these bacterium causes formation of crown gall, isolation, then pathogenic life cycle and the preventive methods. We will study the characterization features, formation of crown gall, isolation, pathogen life cycle and preventive methods in both Agrobacterium tumefaciens and Agrobacterium rhizogene species. The next species of Agrobacterium, which we called as Agrobacterium rhizogene, we will focus on the characteristic feature, that means in what way the Agrobacterium rhizogene differ from Agrobacterium tumefaciens. We will also focus on the genomic and genetic part of this species of the bacteria. We will focus on nutrition and metabolic part. What are the optimum conditions used for the successful culturing of these bacteria? In what way we can isolate this bacteria from the soil, and what are the different processes which we are used in the process of transformation in Rhizobium rhizogene. Then we will study the different current research which are currently right now going on in these different species of Agrobacterium. As we know that both these two bacterium, which belongs to Rhizobiaceae family, that means Agrobacterium tumefaciens and Agrobacterium rhizogenes, these bacterium causes two different types of diseases in plants. That means Agrobacterium tumefaciens causes crown gall formation in the infected plant part which is near to the soil, whereas due to the infection of Agrobacterium rhizogene, there is an hairy root-like structure will form at the infected part of the plants. Once we start to study the introduction of the Agrobacterium bacterium, then we have find out that this bacterium belongs to family Rhizobiaceae, and this is an important gram-negative bacteria which is quite important for biological aspects. This is an rod-shaped bacteria and grow aerobically. This bacteria is present either singly or in the pair. We have find out this bacteria has cosmopolitan host range which affect mainly the dicot plant. This bacterium cause a disease crown gall in the dicot plants near to the roots or stem part near to the soil line as a result of this infection. As we know that Agrobacterium tumefaciens causes crown gall formation in the infected part of the plant. When we focus on the crown gall, that means what do we understand by the term crown gall? How we can identify the specific features of the crown gall in the plant biotechnological experimental studies. Basically, The crown gall, that means they are the tumor-like structure which are connected to the host surface through a narrow capillaries, and they are not easily detachable from the host tissue. There are many tumor-like structures are present on the same plant. It means not only a single tumor is present on the infected plant, but there are many tumor-like structures are present at a same time on the same plant. As a result of this tumor formation, the host plant show their metabolic process. It also changes in their morphological structure. For example, it causes stunning growth. Leaves show chlorotic system, or it show chlorotic necrosis, and sometime once the infection goes very high, in that condition plants goes to be more susceptible under the adverse condition. As a result of this tumor formation, the plant become older, the shape become irregular, and finally the whole plant convert into either brown or blackish in color. That is the isolation of Agrobacterium tumefaciens. Now, in case of isolation, we will select the cells which having the specific plasmid, that is TI plasmid cells, which causes crown gall disease, which should be helpful in the transformation process. Agrobacterium strain lacking TI plasmid gives only these stunning growth in the root part, which is called as rhizosphere inhibiting bacteria, and such part do not cause any type of disease. During the process of isolation, first of all, we isolated the tumor or gall from the infected part, which is the symptom of the tumor or gall in the infected plant part is spongy or crumbling or woody knot-like structures. Once we isolate the tumor gall, we will wash with 18 to 20% of bleach, that is the process of surface sterilization. After the process of surface sterilization, it will be properly rinsed by the sterilized double distilled water many times. Once the process of sterilization of the gall tissue completed, this tissue will break down into the small pieces. Such small pieces put into the buffer solution or the sterile distilled water, vortex it up to 30 to 45 minutes, and then streaking in the suspension medium with the help of inoculating loop. Incubated the medium, then identify the selective strain and detected the such bacterium. Once we start the pathogenic life of this bacteria, we find out that this bacteria live saprophytically in the soil up to 2 years. Host plant is wounded near the soil line, and this bacteria chemotactically move into the wounded part and infected the host cell. Surrounding host cell, stimulated by the infected bacteria, divide rapidly and it form irregular mass of the cell. Finally, this bacterium inserted a piece of its own DNA or gene into the host cell chromosome, causes overproduction of the growth regulator like as cytokinin and auxins. Opines, in this case, opines serve as a nutrient for the pathogen growth. If we consider the preventive methods, then we find out there are susceptible varieties which should not be easily procured by the preventive method. Soil should be planted. In this case, the soil planted with the monocot crops like corns, wheat for the several years, or in case of nursery, we will try to select those plants which are certified from gall-free infection. Or Now these days agrocin 84 is present in market, through which we can prevent the infection of such type of bacteria via soaking of the seeds. Now, in continuity with the Agrobacterium, we come the species of the Agrobacterium, that is Rhizogene, or you can say Agrobacterium Rhizogenes. Recently, this bacterium is called as Rhizobium rhizogene bacteria, which is first of all isolated by Ricker in 1930. This bacteria isolated first time from the rhizogene, which is commonly found in the nodule of the leguminous plant. This type of bacteria causes tumor or hairy root symptoms in the infected plant part. The species name rhizogene itself shows in the Greek word rhiza means root and geno to make root producing bacteria. That means that bacteria after infection causes hairy root like structure at the infected part in the bacteria. The characteristic feature of this bacteria is gram negative. Shape is rod shape like structure, and again this bacteria present singly or in the pair. The optimum temperature for the growth of this bacteria is 25 to 28 degrees centigrade, and in colony morphologically it show convex, circular or smooth in shape. Again, there are different types of strains are available which are used to cause induction, that means which are used to cause hairy root induction in the medicinal plant, and these strains are A4 15834, LBA 9402, etc. Different strain of this bacterium show different types of hairy root inductions efficiency. Induce the formation of proliferated multi-branch adventitious root at the site of the infection. This bacteria causes hairy root syndromes. The characteristic feature of this hairy root infected bacteria or hairy root system in the plant is ability to grow quick in the absence of exogenous plant growth regulators. Hairy root can be obtained directly from the cutting edge of the petiole of the leaf or any explant or the callus. These are the figures which shows the infected leaf by Rhizobium rhizogene species and causes hairy root-like infection or hairy root-like structure in the C and D. Now, when we start to study the genetics of Agrobacterium rhizogenes, then we focus on genome of the rhizogenous then we focus on the genome of the Agrobacterium rhizogenes, which belongs to alpha proteobacteria. In 2014, the complete sequencing of this bacteria takes place by the shotgun sequencing method, and basically the strain used is ATCC15834. That means the whole sequencing of the bacteria in respect to the genome study done on the strain ATCC 158334. Genome of Agrobacterium rhizogene contain 7 lakh base pair in 43 scaffold, which having 60% of GC content. Semi-automated sequencing and automated annotation methods, they are the different tools which are used by the researchers in the study of the genome for the construction of phylogenetic tree using the fast tree 2 software. The whole genome shotgun project was deposited in the gene bank under the accession of JFZP 00000000. It means if we want to take any information regarding with the genome of Agrobacterium rhizogene, we can take information from the gene bank via this accession number. We focus on the nutrition and the metabolic part of this bacteria. We show that this bacteria use oxygen as a electron acceptor. AT plasmid provide the ability in the bacteria to utilize mannopinic acid, agropinic acid as a sole source of the carbon. The optimum condition for the successful culturing of the rhizogene is 25 to 28 degrees centigrade temperature. pH ranges between 5 to 9. Use biotin as a important growth factor. As a result, some opines used by this bacteria as a byproduct which caused by Ri plasmid, and they are the important sources of nitrogen and the energy. We will see how this bacteria infected the plant cell. That means first of all we isolated leaf infected by a suitable bacteria grown in the petri plate, incubated. Finally callus will be formed and this callus will show shoot induction. Once the shoot induction takes place, it will transfer into the rooting medium which later on develop the complete plant. Show Rhizobium transformation process, where we find out that the rhizosphere part, which is the wounded infected part of the plant, secrete some phenolic compound. As a result of the process of chemotaxis, it attracts the bacteria. Under this condition, there are certain genes which are transfer their DNA into the roots of the plant through the wounded infection. After the proper integration and expression in vitro or under natural condition, we will identify phenotypically the study of the hairy root production, which show over development of the hairy root on the surface of the leaf, that is wrinkled root leaf morphology. We will study the transformation of Rhizobium bacteria in two parts, that is A and B. In the figure A, we will find out how Ri plasmid of the bacteria transfer into the plant cell. Here we find out that first of all, plant infected by the bacterium at the part of infected structure, infected part. As a result, some signal molecules are released which should be recognized by the bacterium. These signal molecules help in the attachment of the bacterium with the plant cell. As a result of the infection, the Agrobacterium transfer its plasmid to the plant cell. As a result of the process of the co-cultivation, the bacteria transfer its DNA into the plant genome. Finally, the hairy root-like structure will be developed in the infected plant part. B. We find out how the rhizogene bacteria genetically important for the biotechnological aspects. We find out that Ri plasmid of the Agrobacterium rhizogene enter in the infected plant cell where the chromosomal DNA is also present. As a result of the process of transformation, it enter in the nucleus, and through the process of integration, it transfer its specific part of the DNA into the nuclear genome. Once the proper transformation takes place, we will show or we will study that the hairy root-like structure develop on the leaves or the infected part of the plant, which should be confirmed by the process of PCR. Wild or recombinant type of plasmid causes wounded or wound formation in the infected plant part. As a result, hairy root formation takes place. This is an biotechnological advances of the hairy root of Agrobacterium rhizogenes transfer, which show that in what way this bacterium are important for biotechnological aspect. That is, this will use in the process of phytoremediation. In the it increase the permeability or trapping system of the root. Also used in the scale-up process in the bioreactor. Help in the tromic analysis. Also used in the study of metabolic engineering and other important production of vaccines and the antibiotics. Current stage. If we study the current status of the Agrobacterium species bacteria, the goal is, that is, R100 is the most important strain of the bacteria which successfully induce the plant, that is buckwheat. The goal of this bacteria is to determine the highest growth rate. It increase the root number, improve the transformation efficiency, improve the anthocyanin content, and also improve the very important flavanol content, which is rutin. This bacteria also used to enhance the growth rate of endangered species barbaris, which is commonly called as Indian berry. This is an endangered medicinal plant having important medicinal value due to the presence of important metabolite berberine and an natural alkaloid, which use in the precautions which show an anti-cancerous activity, and this berberine is used in the treatment of diabetes and malaria fever. In this study, the most prompt two different strain of this rhizogene is MTCC 532 and MTCC 2364 for increase the production of berberine. So students, let me summarize this topic. That means what we have learned in this module. Basically, Agrobacterium tumefaciens and Agrobacterium rhizogenes, which having single-stranded DNA and able to transfer their DNA into the host cell and causing different types of disease. It may be crown gall or hairy root-like symptom in the infected part. We have find out that Agrobacterium species show this property due to the presence of specific virulent protein which show their effect in the plant cell. Number 2, DNA transfer initiated when virulence D2 protein nicked border sequences in the tumor inducing plasmid which attach to the 5' end of the gene of the plant cell. Agrobacterium tumefaciens located single-stranded DNA binding protein virulence E2 into the plant cell. Agrobacterium rhizogene strain which lack virulence E2 protein, but it has the tendency to transfer their tRNA efficiently into the gall gene, into the plant cell due to the presence of the gall gene. That in both virulence E2 and full length gall gene contain nuclear localization sequences. These are the specific sequences which help in transfer of single-stranded DNA into the host plant cell. and finally they will start to synthesize the protein in the plant nucleus, in the plant cell nucleus, and finally this virulence E2 accumulated in the nucleus and it pull the transfer RNA into the nucleus of the host cell through the force generating by the cooperative DNA binding method. Gall's full length accumulate inside the nucleus where it predicted that ATP dependent strand transferase enzyme pull this transfer RNA into the nucleus. This difference, this is the main difference, or we can say this is the differensive mechanism for the nuclear import of tDNA, which affect the efficiency and the quality of the transgenics produced by the Agrobacterium tumefaciens and Agrobacterium rhizogenes in the plant biotechnological application. Hence, we can conclude that both that in the agrobacterium, two different species of agrobacterium, the transfer of single stranded DNA takes place with the help of two specific protein, that means virulence E2 protein and Gall's full length proteins, which help in the transfer of tDNA into the host cell and show their efficient method in the host plant cell. Thank you.